In this video, we'll look at how to calculate the frequency of the normal versus ear tuft alleles in chickens. The question reads, the ear tuft allele in chickens is autosomal and produces feathered skin projections near the ear on each side of the head. Now, when we're talking about autosomal traits, we're talking about traits that are encoded by genes found on the non-sex chromosomes. This gene in particular is dominant and lethal in the homozygous state. In other words, homozygous dominant embryos do not hatch from the egg. Assume that in a population of 6,000 chickens, 2,000 have no ear tufts and 4,000 have ear tufts. What are the frequencies of the normal versus ear tufts allele in this population? On your screen right now is a picture of a chicken with ear tufts and one without. To understand this question, let's start by giving this gene a letter. We'll call R the dominant allele and little r the recessive allele. Only if the chicken's genotype is capital R, that the birds don't hatch. But if their genotype is capital R, little r, which means that they're heterozygous, then they will hatch. And they'll live a life with ear tufts. If the chicken is little r, then it will have no ear tufts. So now going back to what's given in the question, we're told that 2,000 have no ear tufts. So 2,000 exist with the genotype being little r. Altogether, these 2,000 chickens have a collective amount of 4,000 little r normal alleles. The reason why it's 4,000 is because each chicken inherits one allele from both parents. And together, that makes 4,000 in total. 4,000 out of 6,000 have the genotype capital R, little r. This means that we will get 4,000 little r's from this group and 4,000 capital R's because their genotype is capital R, little r. Altogether, that makes 8,000 normal alleles and 4,000 tuft alleles. Altogether, that is 12,000 alleles for this particular gene. So to get the frequency of normal, we'll take 8,000 and divide it by the total number of alleles, which amounts to 12,000 in this group of 6,000 chickens. And this will represent the allele frequency of little r. Dividing this out is 2/3, or 66%, of the gene pool consists of little r. And for the ear tuft alleles, we'll take 4,000 divided by the total number of 12,000. That's equal to 1/3, or 33%, of the gene pool for this particular gene is capital R. Now, to check to see if this population is in Hardy-Weinberg equilibrium, we can do a cross of two heterozygous chickens. So let's say that we're crossing a male heterozygous chicken with a female heterozygous chicken. Remember that the frequency of capital R in the population is 1/3, and the frequency of little r is 2/3. And similarly, 1/3 and 2/3. The offspring, or the hypothetical offspring, of these two heterozygous chickens would be capital R, and 1/3 times 1/3 is 1/9. Capital R and little r, multiplying those, you get 2/9. A heterozygous with the genotype capital R, little r, multiplying those, we get 2/9, and little r, which is 4/9. The frequency of heterozygous individuals in the next generation is 4/9. Simply add 2/9 plus 2/9. And the frequency of homozygous recessive is also 4/9. Because these two frequencies are equal and are completely different than the previous generation, it suggests that the population is not in Hardy-Weinberg equilibrium. Notice how it was 4,000 out of 6,000 for heterozygous, which makes 2/3, and 2,000 out of 6,000 for the homozygous recessive, which makes 1/3. And there you have it. That is how to calculate the allele frequencies of a gene and how to check if the population is in Hardy-Weinberg equilibrium.